All right, here's another video. Let's talk about dominance. So far we've talked about something called complete dominance, which, as you can see in this first row here, we designate one of these as dominant, so big P, and the other one is recessive. So in this case, we have big P, homozygous dominance, and this one is heterozygous. So homozygous dominant means we have two of the same allele, and both of them are dominant. But the second case, the heterozygous one, the big P will somehow dominate over the recessive. So you end up with a purple flower, no matter what, because it doesn't matter what the recessive one does. The big P allele dominates over that one completely. So this is what we call complete dominance. Now, there are two other types of dominance out of the three types of dominance that we haven't talked about so far. So this is the new stuff. The first one is called incomplete dominance. So incomplete dominance just means that basically you can have a red type, you can have a white type, and then when a flower has these two different types of alleles, it kind of mixes. So obviously the alleles are genes or parts of are genes that code for different types of proteins. So it may be that when you have CRCR, that produces full red, CWCW, that produces full white. But when you have CRCW, it creates a mix. So a mix of red and white we would think of as pink. So this is called incomplete dominance. That means the two alleles are just as dominant with each other. There's no recessive here. But when the two alleles, two different types of alleles or more, are put together in an individual, in an organism, we have a mixing effect. Okay, so complete dominance is the complete taking over from one allele taking over the recessive. Incomplete dominance is when you have two different types of alleles and they mix. And then the final one is called codominance. And codominance is when both types of alleles get expressed at the same time. So there's no mixing going on. But let's say the A type in this blood type example, the A type codes for this type of protein that shows up on the red blood cell. B codes for this type of protein that shows on the red blood cell. Well, when it's IAIB, then both proteins get produced and both of them exist together. So that's an example of codominance. So subtle difference between codominance and incomplete dominance, but incomplete, usually we see a mixing of things, and codominance, we usually see both being expressed. Okay. So if that makes sense to you, and you can read some of these definitions here, which might help you further because it's got some really good examples. And here's a really good example. If we take a look at this picture here, we've got an example of something that is dominant. Okay, so let's say red color is dominant for this flower. White color is recessive. What happens when you mix them together, if that is a codominant relationship, then you'll see both of them being expressed at the same time. So you'll see red and white being expressed. Incomplete dominance, you will see a mixture of red and white, which makes pink in this case. Okay, so that's a good example. So let's take a look at some problems. Now at this point you can hit pause and go back, and if you don't understand the differences between the three types of dominance, complete dominance, incomplete dominance, and codominance, please make sure you look over the notes and the examples until you do understand, and look at some other videos as well and some other texts online. If you do understand, let's go on to look at some of these problems. So first of all, I should point out, and it says here in this note here, that we use different symbols when it comes to codominance and incomplete dominance. And let me just go back up for a sec and show you the different symbols that we were using. So you can see here that for complete dominance, we used big P, small p. But when it came to incomplete dominance, we used the same letter for both alleles, except we put a little subscript on top to indicate what variation it is. So the reason why we would use the same letter for both alleles is to show that they're both just as dominant, and to show they're both part of the same gene, just different variations. Now to show the variations, we have a superscript, R or W. Same thing with codominance. I represents the same gene on, in this case, blood typing, which is a gene on chromosome 9, and A and B, which represents the two different variations. So for incomplete dominance and codominance, we show the same letter for that gene and then superscripts indicating the different alleles. That's how we can tell by looking at these symbols whether we're talking about incomplete and codominance or we're talking about complete dominance, in which case we just use the letter, no superscripts. Okay, so let's go back down to the question and let's work on this problem together. It says, in humans, hair type is determined by incomplete dominance. So that's important to highlight. That means we're going to use the special symbols that we just learned. And also we're going to realize that when you combine the two different types of alleles, two variations, it's going to have a mixing effect. So it says here, some people have curly hair, and it gives you the genotype, HCHC. Some people have straight hair, genotype HSHS. And heterozygotes have wavy hair. So you can see wavy hair is where you get one HC from a parent and the other is an HS from the other parent. And so you have kind of an in-between between curly hair and straight hair, which is wavy hair. It says, the question is asking, what would the probability distributions be if a person with wavy hair had children with a person with straight hair? So, as usual, let's write out what the cross would be. So, you're mating somebody with wavy hair. So, wavy hair would be HCHS. You're crossing that person with somebody with straight hair. Straight hair would be HSHS. Okay. So you're crossing wavy hair with straight hair. Let's double check to make sure you got both of those correct. And then we're going to draw a Punnett square. So I'm going to draw a Punnett square. And it doesn't matter which parent you put on the top. Let's just go with the wavy hair parent. So HCHS. And then off to the left side, I'm going to put the straight hair parent, HSHS. Now we're going to cross them. So our offspring would be HCHS. HCHS, HSHS, and let's just make sure we keep the letters in the same order. So let's, I don't know, let's go with alphabetical. So we'll put this as HCHS. Otherwise, if we put this last one I just did as HSHC, that one and the first one might look different, which they're not. So we're going to make them both HCHS. And this last one here, HSHS. So it's asking, what would the probability distributions be? Well, we have two ratios to write down. This is the genotypic ratio. So for the genotypic ratio, we have 1/2 HCHS and 1/2 HSHS. For the phenotypic ratio, we're going to have 1/2 is going to be HCHS. HCHS is between straight and curly, which is wavy. So we're going to write wavy. And the other half will be straight. So half wavy, half straight hair. Okay, let's move this up. It says, what ratio of offspring would result if a person with wavy hair mates with someone else who also has wavy hair? Okay, so let's take a look at what the cross would look like. So wavy hair is HCHS crossed with HCHS. And let's draw the Punnett Square. HCHS is one parent, HCHS is the other parent, and when we cross them we get HCHC in this corner, HCHS over here. Again, let's keep it in alphabetical order, so HCHS, and then this one here, HSHS. So the ratio of offspring, genotypic. The genotypic ratio of offspring looks like we have 3 different types here. So the first type, so 1/4 would be HCHC. Half would be HCHS, and the half that I'm referring to are these ones here, HCHS. Okay. And the last quarter, HSHS. Let's do the phenotypic ratio. Phenotypic ratio, we have also 3 different types, because we know that HCHC is curly, so quarter is going to be curly. We know HCHS is going to be wavy, so half is going to be wavy, and then the last quarter here is straight. All right, so you're going to have 3 different possibilities of phenotypes, because as we explained, for incomplete dominance, both are just as dominant, but you have a mixing effect. So between curly and straight hair, and in this case, in this problem, it's wavy. Okay, let's go on to the next question, which is about codominance. It says cattle can be red, which is HRHR, and white, or something called roan. So roan is growing both red and white hair at the same time. So kind of like a spotted or patterned cow of some sort with both colors. It says predict the ratios of the offspring when the homozygous white cow is crossed with a roan bull. So homozygous white cow, let's draw the cross or write down the cross here. Homozygous white cow, that's a homozygous white cow, is crossed with a roan bull. A roan bull is a hybrid, so HRHW. That's our cross. It says predict the ratios of the offspring. Well, you have to do a Punnett Square. Again, it doesn't matter which parent you start with at the top. Just choose one, and the other one goes down the left side. Okay. Let's start with this. HRHW. Let's put this in the same order. HRHW. HW, 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 HW. Okay, so the ratios. Genotypic. The genotypic ratios. We have 2. So one of them, so half of it, 1/2 would be HRHW. The other half would be HWHW. And phenotypic. Half is called roan. And then the other half would be white. Okay, so this is different from incomplete dominance because we don't mix the white and the red to make a pink cow, although that would be really cool. In this case, we have codominance, which means that both red and white both show up in the phenotype at the same time. So that's the slight difference between codominance and incomplete dominance. What ratio of offspring would result if 2 roan bulls are crossed? Okay, let's write out the cross again. So we have roan is HRHW times another, or crossed with another roan bull. Okay. I just realized it says 2 roan bulls. I guess one of these need to be a cow. Anyways. So for crossing these two, then we're going to draw the Punnett Square. HR, HW, HR, HW. Here are the resulting probabilities for the offspring. Now I'm getting lazy. Genotypic ratio. Looks like it's going to be a quarter, red. Was it red? Huh. Quarter red bull. And we've got 1/2 roan. And then 1/4 white. Oh, of course, that's not the genotypic ratios. So it's not a genotypic ratio at all. Genotypic ratio, a quarter HR, a half HW, and a quarter HW. Now, phenotypic ratios should be a quarter red, half roan, and a quarter white. All right. Now it says here the most relevant use of codominance, or most relevant for us where it appears, is blood types. So there's actually 3 types of alleles when we're talking about blood type. And it shows in this chart here. So there's actually, for blood type, there's IA, IB, or little I. So there is a recessive allele here. So if you are type A blood, that means you might have IA from one parent and IA from another parent, or you could be IA from one parent and then the type O recessive from the other parent. Okay. Type B is just as shown here. We've got IB or IBI. Type AB is this is where it's incomplete dominance, or sorry, codominance, where you have A and B showing up on the red blood cells. And then type O is little I. So when you have little I, your type O blood, which these all refer to different proteins that show up on the surface of your red blood cells. So for type O, you don't have those particular proteins showing up. Okay. So let's take a look at this question. It says, what ratio of offspring will result from a man with type A blood who is heterozygous for blood type, so type A, heterozygous, and a woman with type B blood who is also heterozygous for blood type, so type B, heterozygous. So let's write out the cross. So a man, and for blood types, we always use the letter I for that gene. For a man with type A, but he's heterozygous, so that means the other one is not IA. The only other possibility is it's a small I, heterozygous. And the woman happens to be type B, and she is also heterozygous for blood type. So that's going to be your cross. So we'll draw our Punnett Square. Here are the possibilities. Okay. Ratio of offspring. Genotypic ratio. We've got 1/4 IA, IB. We've got 1/4 IBI, 1/4 IAI, and then 1/4 little I. Let's look at the phenotypic ratio. Okay, so phenotypic ratio, we would expect 1/2 to be type AB blood, 1/4 to be type B, 1/4 to be type A, and then 1/4 to be type O.